Good morning, everyone. I hope you can hear me. Welcome to this webinar on tackling key pests in UK horticulture. I'm Natalie Key, Knowledge Exchange Manager for AHDB Horticulture. And just before we kick off, I wanted to run through the order of play for the webinar and some housekeeping elements. So first of all, we'll hear from James Phillips from BBSRC, who will provide some background to the pilot initiative between AHDB and BBSRC, which is the rapid response to high priority pests and diseases in UK horticulture, which funded all the projects you're going to be hearing about. We'll then pass over to Professor William Kirk, who will be discussing his project looking into the development of their technique to identify pheromones to monitor and trap thrips. We'll move on to Dr. Glen Powell, who will then talk about his project developing a rapid identification method for brown marmorated stink-bug. And finally, Dr. Michelle Fountain will discuss her work identifying the sex pheromone of Nesidiocoris. So just for some housekeeping, for information throughout the webinar, the audience is muted, but you can ask questions by submitting them through the questions box. And there'll be opportunities to ask questions after each session and also at the end. So ideally, please also indicate who the question is for. Any questions that we don't get to will be saved and answered where possible after the webinar. Similarly, if you have follow-up questions once we're finished, please send them to my email, natalie.key@ahdb.org.uk by the end of next week, and we'll do our best to answer these as well. The webinar will be recorded and made available on our web pages, so you'll be able to catch up with content after the event. If you have any problems during the webinar, please either let me know through the questions box or email me again, that's fine too. So without further ado, I think that's all I needed to cover, so pass over to James to provide us with a bit of an introduction.
Excellent. Thank you, Natalie. So hopefully you can see my screen now, if that's okay.
It looks good to me.
Perfect. Brilliant. So thank you very much for joining us today. Really excited to talk about this new initiative between BBSRC and AHDB. So I've just got a few slides just to introduce today, basically. So let's just see if I can get this to work. There we go. Great. So if you haven't come across this before, BBSRC is the Biotechnology and Biological Sciences Research Council, one of the bodies that make up UKRI, which is the UK's national public funder of research and innovation. BBSRC invests in bioscience, and we're particularly interested in issues around food security and how we can support the agricultural and horticultural sector. So for this initiative, we teamed up with our colleagues at AHDB to invest £250,000 to support 5 projects for about 3 to 5 months to tackle some of the most urgent and high priority pests and diseases facing the UK, which present a significant and immediate threat to UK production. The targets were identified through an exercise led by AHDB with their grower panels as part of a wider piece of work around maintaining the UK National Risk Register. The research itself is investigating fundamental biology, pests and diseases, and hopefully will stimulate all sorts of interesting further research that will redevelop and deliver benefits to the grower community. So here's a list of the excellent projects that we funded, and we received lots of fantastic applications, so thanks again to the community for supporting the process. Looking forward in a minute to hearing from some of the presenters and the leads of these projects. One of the crucial things about this is that it's really one of the first times that AHDB and BBSRC have worked together in this way to support the community. So hopefully it's the start of more of that kind of cooperation. We're looking forward to working together with our colleagues in AHDB more in the future. Finally, I just wanted to finish off with a quick note. Obviously, things are a bit unusual at the moment with COVID-19. But we are still very much open for business. We're still accepting applications, and in some cases, some of our application deadlines have been extended to give people longer to apply. But if you are interested in working with academia, please do send me emails after this call. Very happy to talk you through the different schemes that we offer, including our industrial plant awards and BBSRC link programs. So that's the final slide from me. Some contact details there. Very happy to take questions later on. Thank you.
Thank you very much, James. So now we'll hear from William Kirk.
Okay. Rachel, can you hear me and see me? Natalie.
Yes, yes,
Sorry.
I can hear you and I can see your presentation.
Right.
That's okay. Go ahead.
Let me just try and get this onto full screen. Can you see it now? So it should be a bit bigger.
Perfect. Yep.
Okay. Good morning, everyone. My talk is on the identification of pheromones to trap thrips, and the project behind it had a somewhat longer title. It was development of a solid injection technique to identify thrips pheromones for monitoring and mass trapping in UK horticulture. And I led the project, and my co-investigator was Falco Dryfout, and we both at Keele University. As you can see from the title, it was a techniques project. So we were developing techniques to find ways of identifying thrips pheromones that work better than the methods we've had previously, which means a lot of it is techniques and therefore probably not terribly interesting. So what I'm going to talk about is a bit of a background to mass trapping, and then really skip the actual detail of what we did, but tell you where we've got to at the end of it. So these are the culprits. These are thrips. This is a western flower thrips. They're a big problem in horticulture and agriculture pretty much around the world. They're very resistant to insecticides. They're very hard to control. They hide away, so they're difficult to get at them, and they breed very fast. So they pose enormous problems. In addition, there are several new species coming along, so they keep, once you think you've solved one problem, another one comes along after it. So it's an ongoing problem. And the sort of damage they cause is either, as you can see this kind of on the left, cosmetic damage to ornamentals, which makes them unsaleable, or on the right, a sweet pepper, you can see it's stunted, scarred, shrunken, again, pretty much unsaleable. And the damage can be done very early when you can hardly see the fruit, and perhaps not even know the thrips are there. So it's sometimes quite a cryptic problem initially, and the damage can be done early. Besides actually damaging the crops, they can also transmit plant viruses. Many of the pest species can do this, and a group called tosper viruses. This is a picture from California where you can see along the table sort of alternating ones damaged by virus and then healthy ones. I think you can probably clearly see the difference there. Right at the end of a table, there are sort of shrunken, scarred, deformed peppers and some very nice healthy ones in front. And we don't have too much of a problem with tosper viruses in this country, but it's a very big problem in the USA and China in particular. So damage to crops by feeding and transmission of viruses. One problem we had some time ago was establishing whether trapping could be effective. We use trapping for monitoring, certainly, and often use blue traps to trap the thrips, but would traps be effective enough? Would they trap enough to actually reduce the population size? So this is the result of an experiment from a previous project, which was funded by AHDB. And if I take you through this is an experiment done in a UK strawberry crop. And along the bottom, you can see for 3 months, July, August, and September. Let me just use my pointer here. Okay, so July, August, and September, 3 months, and we had 3 treatments. One was no traps, which was just using the grower's normal controls, which involved biological control with predatory mites, repeated releases. And then the blue treatment here was blue sticky roller traps across the crop. And the red treatment here was traps with the pheromone produced by Western flower thrips. And across down the side here, you can see the damage we measured from bronze seeds, so the amount of damage that was done to the strawberries. And then, across here, you can see the damage threshold. So if the damage to the seeds exceeded that threshold, as you can see here, then the strawberries were being downgraded in the packing house and the grower was losing money. So what happened is that in July, the grower's normal control methods were working. In August, well, not working quite so well, but by September, the predatory mites had rather lost control and significant damage was occurring. But where we had the mass trapping with the traps, either without the pheromone or with the pheromone, we were reducing the damage significantly. So this showed quite clearly that mass trapping could work in UK strawberry crops. I should perhaps point out at this stage that traps can be sold and used for monitoring, but at the moment they're not approved for mass trapping. Okay, so we established the principle that mass trapping works and that pheromone can be useful. And in fact, there are several products using the pheromone for Western flower thrips. You can see here products involving septa, which can be stuck on traps to increase trap catch. And here's another type of septum from another manufacturer. Another product that's produced is a roller trap with pheromone, Western flower thrips pheromone impregnated into the glue of the trap. So these can be unrolled and can increase the trap catch. So products available, we know what the Western flower thrips pheromone is, but it was very difficult to identify. There are other pest thrips which are a problem in UK horticulture and causing concern. So for example, the onion thrips, we don't know what the pheromone is for that species. It's a big problem in onions and leeks, but also in many glasshouse crops. The rubus thrips, Thrips major, and the rose thrips, Thrips fuscipenis, these are both species that are becoming increasingly of concern to strawberry growers and perhaps some other soft fruit growers. Poinsettia thrips, which is Echina thrips, that's a problem in some glasshouses. And then there's another species that's recently arrived, the Japanese flower thrips, Thrips cytosus. Despite the name, that's more of a problem on leaves of crops, and it's quite prolificous. It feeds on very many different plants. And I put at the bottom, other invasive thrip species arriving soon. So we've got quite a list where we don't know the pheromone, and we've got other ones, and we want to be able to identify the pheromone quickly if these other ones do arrive. So where we'd got to at that stage is we could identify the pheromone, but it was very difficult. And what we wanted was a better technique for doing this. So the previous methods, difficult because thrips produce so little pheromone, they need to we need to collect very large amounts. So if I go through these sort of the bullet points, the way to do it is you need to separate out live thrips of one species. So in a crop, there's often a mix of species and you can't have the wrong species contaminating it. So separating species of thrips from the live specimens is quite difficult and tedious. You would probably need about hundreds or thousands of live males to be able to collect enough pheromone. So that in itself makes the whole thing very difficult and only really possible for a few pest species that are present in very large numbers at one time without any other species present. And the techniques that have been used before are SPME fibers. That's one technique, but the problem with that is you cannot transport it from far away because the fibers lose the pheromone very quickly. And another technique is using something called Tenax, but you need a lot of thrips. Tests to identify the pheromone in the past have used hundreds, if not thousands sometimes, of thrips. And again, that's very difficult to obtain live and separate them out. So the problem we faced was how could we develop a technique that could do this much more effectively with far fewer thrips. So that's a technique that's been used in the past. You can see here these little tubes. It's a close-up. We can have this SPME fiber and you can put 100, 200, 300 thrips in there and try and collect enough pheromone. But as I said, it's really very difficult and you can only do it if the the thrips on the doorstep of where you do the analysis. So this is what we've identified previously. This is the Western flower thrips pheromone. The way we do this is to take a very large number of males, and you can see these traces here. This is from what's called a gas chromatograph. All the different peaks represent all the compounds that are separated out. And what we look for is a compound that's produced in the males. So this upper trace and these red arrows show compounds that are particularly present in the males. And then the trace below shows the females. And so what we're looking for is something that's present in the males, but there's no matching peak in the females. And these were the two compounds we identified back in about 2000, that are the pheromone of the Western flower thrips. But since then, across the world, only 4 or 5 species have had their pheromones identified, mainly because of this difficulty of the technique. So what do we try to do? Well, we wanted to find out, could the pheromone be extracted from dead males? If they produce enough and have it stored, then there's enough male pheromone within the males, and we could collect up the males and accumulate them and get enough pheromone that way. So that was one question. And then the second question was, if we can't get pheromone from the dead males, could we do a more sensitive technique that just needs a fewer live males and doesn't need hundreds or thousands? So now if I cut through the 4 months of work, which the project ran from start of November 2019 to the end of February 2020, and tell you actually something about where we got to. We developed this technique, this is a solid injector, so we can collect the thrips. And if I move my cursor over here, and my dashboard is in the way. So here you can see a tiny glass tube that has thrips inside it. It's inserted into this inlet. The glass is heated up and crushed, and that drives off the pheromone into the gas chromatograph. So that's more sensitive because we don't need a solvent to do it. And if we combine that with a very much more sensitive GCMS, this is called a gas chromatograph time of flight mass spectrometer. These are phenomenally expensive. There aren't many of them in the country, but that is much more sensitive to pheromones. So we did a combination of how we handle the thrips to collect the pheromone, how we collect it, how we inject it, and the type of machine we use. And so after playing around with this, trying all sorts of possible things for 4 months, we ended up with a technique that worked better than the previous technique. So our conclusions at the end, let me keep track of time, not too bad. Our conclusions at the end was that the males do not produce sufficient pheromone for it to be extracted from dead males because they don't store it. In other words, they only produce the pheromone when they're attracting a mate. So perhaps I should have mentioned that males produce the pheromone, but it attracts both males and females because the males aggregate to produce a kind of mating swarm, and the females come to that. So the advantage of this pheromone is it attracts males and females, which is what we want. But then they don't store it, so we couldn't use the dead males. That's a bit disappointing, a bit of a setback. But then we moved to the second stage, which was developing a way of collecting it better and a more sensitive detection. So we now have a method that can entrain enough pheromone for identification from only 10 live males at a time. Now that makes the whole thing much easier. Collecting 10 live males is not too difficult. Collecting 1,000 is much more difficult. Now that we managed to get there at the end of the 4 months, but because it was the middle of winter, we couldn't easily get thrips then. So we have a colleague in Hungary, and unfortunately he started to be locked down just when he was about to send the specimens. We managed to get some specimens. They've reached me, I've got them in my freezer, but we can't analyze them yet because we have to, our labs are closed. So the state we're in at the moment is that we've developed a technique that we think can identify pheromones fairly quickly and easily, but we're held back. So disappointingly, we weren't within the project able to identify a particular pheromone, but we think we're well placed to do it in the future. And I'd just like to acknowledge Falco Dryfout, who was the co-investigator on this project, and Dr. Anka Kovac, who was the postdoctoral researcher who did most of the research. Thank you very much.
Thank you, William. We have one question for you.
Okay.
So regarding the Thrips pheromones, are these sex pheromones? And he continues to ask, when only trapping males, mass trapping often doesn't work in other crop or pest situations. I noticed that the mass trapping results presented, the pheromone didn't actually increase trap catches. So I guess the initial part was if it was a sex pheromone.
Right, so it's technically called an aggregation pheromone. The sex pheromone is produced by one sex that only attracts the other sex. So if a male's produced and only attracted to females, we'd call it a sex pheromone. But in this case, as I said earlier, it attracts both males and females. So it's called an aggregation pheromone. We know it increases the trap catch. It doubles or triples the trap catch on a blue cat trap, although it wasn't significantly decreased in the graph I showed you. That had rather few replicates. We know from counts directly on the traps that it was doubling the trap catch. So we know that reduction was a real reduction.
Okay.
Was that all the questions? I think so.
There's one more from someone else. Quick question, how were thrip population levels measured or estimated for the trap efficiency experiments?
Right, it was done in 3 ways. One by examining the flowers by eye to count the number of adults in the flowers. It was also done by counting the numbers that were caught on the traps. So the graph I didn't show you because I'd only got 15 minutes. We have another similar graph showing that the density of thrips in the crop had gone down, and the graph looked very similar. So we got a big reduction of thrips in the crop, but the interesting graph was the one at the end, which often experiments don't get to, which is showing that there really was a reduction in damage. And in fact, that we did a cost-benefit analysis. The cost of the trapping was cost-effective when you relate it to the saving in damage. So yeah, so the answer is we counted thrips in the flowers, we measured the thrips on the traps, and we measured the damage done to the fruit.
Great, thank you. A couple more questions. What species did you get from Hungary?
Thrips tabassi, which is the onion thrips, which is the one we think is of probably most widespread interest. It was the only one we could get because we had to have something that was being cultured in December. Very few species of a pest significance are cultured. However, perhaps I should just point out that AHDB are just funding a project which is just starting, where we're going to try to culture some of these other species, and that can get us enough numbers of just the one species. And perhaps also mention that Jude Benison, Peter Seymour and I produced a review of these other species for AHDB recently, and I'm not sure whether that's actually come out and available to growers. It may be out now, or if it's not, it should be very shortly.
Okay, I'll have a look into that and see, and can feedback. Once identified, is it very difficult to produce the pheromone on a commercial scale?
It depends what the pheromone is. I think perhaps the thing to say, if you produce a large amount, let's say you produce a litre of it, it would cost quite a bit, but the amount you use is extremely small. We're talking about micrograms on a trap. So it's not that expensive because of the very small amount that's needed.
Okay, thank you. And one last question for you. What species of thrips feeds on brassicas, do you know?
On brassicas, what do you think of something like oilseed rape or
they haven't
cabbage?
Specified. Yeah, but I would imagine
it depends. Cabbage, there's a species called Thrips angusticeps that's often a pest on cabbage seedlings.
She's clarified oilseed rape. Yeah.
Oilseed rape. Okay, nothing breeds in very large numbers. You get quite a range of different species. You might get Thrips angusticeps, Thrips tabassi, many of the very prolificous species which are pests like Thrips major, Thrips fuscipennis. You also get a predatory thrips, the black and white striped thrips, an Eola thrips, and that's actually beneficial because that feeds on the other thrips.
Thank you very much.
Okay.
Yep, I think that's don't have any further questions. So thank you very much, William. We'll pass on to Glen.
Okay, I'll just start by checking that you can hear me and see my screen. Is that okay, Natalie?
Yeah, looks good to me. Thank you.
Okay, thanks. Well, good morning, everyone. I'm reporting on a similar short project, which was done in collaboration with Beth Clare at Queen Mary University of London. And we aim to develop a DNA-based test for the invasive pest brown marmorated stink bug, Haliomorpha halis. I'll summarize the work, but before I do that, I'll just give some background on this stink bug as a pest and what we've been doing at NIAB to monitor for it. So stink bugs, or shield bugs as we tend to call them in the UK, have a life cycle where they produce an egg mass, and then when the nymphs hatch from the egg, the first instars tend to kind of cluster around the egg mass, and then the subsequent stages disperse and walk away, perhaps onto other plants. There are 5 nymph stages in total before they molt finally to adults, and then the adults, of course, can fly and disperse over greater distances. So all that's pretty standard for any shield bug species. But brown marmorated stink bug has particular characteristics that make it very high impact as a pest. And one of these is that it has a very broad host range. So it can feed on hundreds well, over 100 different plants, lots of different crops, including tree fruit, soft fruit, and grapes, vegetables, etc. Apples and pears are probably at most high risk in terms of the UK situation. It's also sort of globally important, and another feature of this bug is that it has a very high capacity to disperse, not only through flight when it arrives somewhere, but also as a hitchhiker. It has a tendency to overwinter in large aggregations, and so the adults will cluster together in the autumn, often inside static structures under tree bark or on the edges of buildings and so on, but also inside packaging, inside products, and inside machinery as well. So as all these things are exported around the world, the bug tends to move with them. And so this is the kind of global situation, or at least it was a couple of years ago when this review image was published. The sort of native range is Southeast Asia. The bug arrived in the USA in the mid-90s. It subsequently also appeared in Europe, initially in Switzerland in a few years leading up to around 2007. And subsequently it's appeared in other countries in Europe, in Canada, in Russia, and in the Southern Hemisphere 3 years ago in Chile. The situation in Europe is shown here in a bit more detail. This is a slide that's been shared by our collaborator in Italy, Laura Maestrello, and she's been mapping the appearance of the stink bug in Europe. And as I say, it arrived initially in Switzerland, kind of the mid-noughties, and then sort of also was reported in other regions. It's now particularly prevalent in northern Italy, where it's caused extensive damage to crops, but is also very abundant in other countries, including Hungary, Romania, and Georgia. This is not the most up-to-date picture, because if we look at the UK, then these sort of crosses with circles show interceptions, and the stink bug has been intercepted actually on several occasions by inspectors examining passenger luggage and imported products and so on. But as I'll show in a few slides, we do now have evidence that there are live bugs being found in the UK as well. In terms of impact, the impact on crops is extensive, and as I said, it does have a very broad host range. They have, like other sap-feeding bugs, they have the stylet mouthparts that they use to sap the juice from plants, and they particularly favour growing parts such as flowers and developing fruit. And they inject toxic saliva, which leads to the development of these brown lesions. It also has an impact through an urban nuisance sort of factor as well, because of that overwintering kind of aggregation in the autumn. That behaviour, where the bug has high populations, leads to homeowners being sort of bothered by large clusters of the insects on exterior surfaces, but also entering the houses as well. In terms of management options, there's been quite a lot of research done since this sort of global pest has been spreading and causing problems for a number of years. And in terms of the efficacy of insecticides, they're actually difficult to kill. Even if they're knocked down, they can sort of recover again. And it tends to be the broad spectrum products such as pyrethroids that are most effective, unfortunately. And so obviously it's difficult to incorporate those types of products effectively in pest management in an integrated way. But there are smarter ways of applying these than spraying the whole crop. And they can be combined with the aggregation pheromone, which has been synthesized and is available to attract and kill the pest, either spraying baited, pheromone-baited trees or nets. Netting has also been used as exclusion netting, and that's being deployed particularly in Italy to protect peaches and nectarines and apples, but it's obviously rather expensive to install. And also there's a lot of research interest in biological control. Unfortunately, generalist predators are not terribly effective at controlling brown marmorated stink bug, but egg parasitoids, these tiny parasitic wasps that can lay their eggs inside the eggs of the stink bug, are of particular interest. And this species, Trisulcus japonicus, the so-called samurai wasp, is from the same kind of native original range as brown marmorated stink bug, so may have good potential for biological control. So just to summarise what we've been doing at NIAB, we've been doing some surveillance activities to try to survey for whether the stink bug is present in the UK, so that we can be alerted and alert growers at an early stage when it becomes established. And there have been three sort of activities connected with surveillance. One of these has involved pheromone monitoring using pheromone-baited traps. So these are the commercially available high-dose pheromone lures that are available from the USA. And those are used in combination with double-sided sticky traps. And these traps have been shown to be effective where the stink bug is present in other areas of the world. So we've actually put these traps out in southern England in 2018 and 2019 at 10 sites in each of those years. And the traps are set out, put out at sites for a 24-week period and checked every week through the summer and autumn. The yellow dots show the sites in 2018, which are mostly clustered in Kent, but also up in Cambridge and Suffolk. And last year we also had sites, some of the same sites, but traps also across in Berkshire and also one in Wales. And the sites were a mixture of grower sites in orchards, but also botanic gardens at the University of Cambridge, the Natural History Wildlife Garden in London, and a campus garden at the University of Reading, because the bug often becomes established initially feeding on exotic trees in urban centres, and we know that from the experiences of other countries. So I can summarise very, very quickly the results of all this is in that we have not caught brown marmorated stink bug in the UK so far using these pheromone traps. So that's good news, but also it's good news that we have not caught other native UK shield bug species, which also would have aggregation pheromones, and sometimes these pheromones can be very similar between different species. So it's good that we're not getting bycatch of the native species that might sort of interfere with any future trapping programs. Another approach has been to do surveillance based on internet searches and following up by contacting people to see and follow up reports of brown marmorated stink bug in the UK. And an adult was reported in Hampshire in November 2018. This was actually identified by a pest control specialist who was emptying a rodent bait box at a property and found a hibernating brown marmorated stink bug adult inside there and recognised it, and that has been confirmed. Also, looking at the amateur naturalist app, iNaturalist, there are a couple of red squares on the map for brown marmorated stink bug, and particularly interestingly this one, which was reported in Hampshire very close to the site of the other bug just 6 months earlier. So these two adults could just be coincidentally quite close together, or might reflect the beginnings of a population. There's this second adult reported in Cheshire very recently, but that actually seems to have been imported in some packaging material with some imported fruit. So I don't think that one is quite so significant, but the two adults in Hampshire are significant because they were free in the environment when they were found, rather than obviously associated with any imported material. And then the third and final thing that we've been doing as part of surveillance is actually publicising the fact that we're interested in this invasive pest and inviting people to send us images or even better specimens of bugs that are found and thought to be brown marmorated stink bug. As you can see, we've had several of these images and specimens sent to us. However, none of those have been brown marmorated stink bug. They've all been either these two, which are native shield bug species, or these two, which are the western conifer seed bug and mottled shield bug, which are other invasive species. So I think what this is highlighting is that it's very easy to get confused between brown marmorated stink bug and other shield bug species that are present in the UK. And that's with the adults. It's even more difficult when you're looking at other life stages, nymphs or egg masses. If these are found, then it's even more challenging to identify them. But we could be looking at fragments of egg masses or fragments of insects that are found on sticky traps. And so that's really why we need a reliable diagnostic test. So the new project, the collaboration with Beth Clare at Queen Mary, aim to develop a high throughput method for detection of brown marmorated stink bug and to evaluate the reliability of the method. The basic approach is shown, summarised in this diagram. It involves collecting the sample, doing DNA extraction, and then using primers to PCR amplify the DNA. Now, there was a lot of the project, a lot of the time and effort went into designing effective primers. Initially, we assembled an extensive database of DNA sequences, both for brown marmorated stink bug and for other species that are likely to be confused with brown marmorated stink bugs. So this was thousands of sequences. And based on that, we designed PCR primers with a very high sensitivity to amplify brown marmorated stink bug DNA, but not the DNA of other species. And then the fragments that are amplified are used through high throughput sequencing to identify the sequences against reference databases and identify the species that are present in the sample. So the key outputs of this work were that the specific primers that were designed were shown to be very effective at selectively amplifying the DNA of brown marmorated stink bug and not other species. We showed that this was true not only when we kind of extracted good quality DNA from fresh samples, but also when we tried to use more degraded material, for example, old egg masses that were stored for weeks or months in non-ideal conditions. We also showed that the test works even when the DNA of other species is present. So we deliberately spiked the samples with the DNA of other species, and we still got good detection of brown marmorated stink bug DNA. And we showed that the test was extremely sensitive. We could dilute the DNA of adults, nymphs or eggs, even as much as 10,000 times, and still get PCR products using these very sensitive primers, which you can see here as these bands. So at the end of the project, we made the methodology available, but also made a test available initially at the Queen Mary University of London labs. So that's a link there where samples can be processed on a sort of case-by-case contract basis initially. But we're also in discussion with one of the leading UK-based eDNA companies. So they are specialists in looking at environmental DNA and using that as a basis for monitoring the diversity of species. And that could provide a platform for commercialization and as a high throughput method, and basically a diagnostic test that could be made available to growers. So just in conclusion, we know that brown marmorated stink bug is present in the UK, not just coming in on imports, but it can be found, or it has been found in a few cases as free moving adults in the environment. I think it's important to say that establishment of population, to actually demonstrate that, we would need to find other life stages. So until we find nymphs or egg masses, we can't be sure that the pest is actually established in the UK. Unfortunately, some of our native shield bug species or other invasive species tend to be mistaken for brown marmorated stink bug. And so there will continue to be false alarms and misidentifications in the future. But hopefully the new DNA-based tests will help with that. And I think all this highlights the value of continued future monitoring. So I hope that we'll get funding to continue using pheromone traps to monitor for the pest, but also that we'll be able to further validate and commercialize the DNA-based identification test. Just like to finish by thanking various people. Michelle Fountain, who leads the TF223 project, which is the project that we've been actively surveying for brown marmorated stink bug. Also the BBSRC and the AHDB for funding, Joe and Rosie for the technical support, and our collaborators in Italy, Switzerland and Canada for all their support with information and images and samples that they've sent us, and all the people that have monitored pheromone traps. Okay, that's it. I'll pass back to you, Natalie.
Thank you very much, Glenn. We've got one question in so far, so if anyone has any questions, please submit them now. You mentioned the parasitoid wasp with the same range as brown marmorated stink bug that could be used for biological control. Would you anticipate any adverse impact if these were used in the in the UK if the pest becomes established?
Yes, I mean there has been work done both in Switzerland and in the USA to try to get support and to investigate the potential impact of releasing that species on the native shieldbug species. So I think that particular species is quite good at, in terms of being fairly specialised for brown marmorated stink bug. While that work has been continuing in quarantine sort of facilities, this parasitoid, Trisulcus japonicus, the specialist parasitoid from the native range, has actually turned up in North America and also in Europe. So we don't quite know how, but it seems to be spreading with brown marmorated stink bug, which could be good news in terms of long-term control and suppression of brown marmorated stink bug numbers.
Okay, thank you. What is the economic cost to tree fruit growers where the pest has become established in places such as the USA?
Yeah, so I've been looking at this. So there are a few figures out there, and they tend to be quite a few years old. But for example, in the North Atlantic states, after the arrival in the 90s of brown marmorated stink bug in the USA, in 1 year, 2010, it was estimated that there were $35 million worth of losses to apples alone. So, you know, that's obviously very, very high losses, but not particularly recent figures, I'm afraid. More recently, our colleagues in Italy are seeing losses in terms of more than 300 million euros of losses to apricots, nectarines and peaches. And that's just not the whole of Italy, but just one region, Emilia-Romagna region. So, you know, it can have an extremely high impact.
So it's important for us to try and be prepared.
Absolutely.
So another question, you mentioned double-sided sticky traps for the monitoring. Is a certain colour of the trap needed?
I believe in terms of sticky traps, I think the clear ones are effective, and they are the ones that are provided as standard by North American companies are clear double-sided sticky traps, and those are the ones that we've been using. Trap colour, I think, can have an effect, and there's been a lot of work done on designs and appearances of different traps. In terms of trapping, other designs such as pyramid traps, where the bugs actually enter and climb up into the trap, have been shown to be more effective than sticky traps, although obviously they're more expensive to buy. And black pyramid traps in particular are particularly effective. So colour of trap can be effective, but as far as monitoring using relatively affordable traps that are shown to be effective in detecting the presence of the bug as a sort of sentinel trap, the double-sided sticky traps that we use are thought to be good for the job.
Thank you. What time of year was the monitoring programme taking place in the UK? And I guess in combination with that, there's another question. Has any monitoring been conducted in the north of the UK, or are there plans for that?
Okay, yeah. So for the first part of the time of year, so for our programme, we start at the end of May, early June, because we know that we're going to run for 24 weeks because the high dose lures have been shown to be effective for up to 12 weeks. So we change them after 12 weeks, and then we have a second 12-week period. So we're looking at sort of beginning of June through to kind of November time for our monitoring. After about November, then it's unlikely that any adults around would be actually active and moving around in the environment. They would be tending to hibernate. In terms of the further north in the UK, yes, our colleagues in Scotland have recently run a trapping program. I think that's finished now. I think it was just within one year of funding, but that was involving colleagues at James Hutton Institute and other institutes within Scotland. They were using the same suppliers and designs in terms of the lures and the traps that we're using further south, and we felt it was important to share what we're doing with them so that we could sort of compare our findings and hopefully use the same methodology. As far as I know, no brown marmorated stink bug were found in Scotland. I hope both our monitoring and monitoring further north can continue, but I'm not aware of whether the funding is available to do that at the moment.
Okay, just a couple more. I think we can squeeze some more in. Is the brown marmorated stink bug established in countries that have a similar climate to ours?
Yes, that's a good question. There is establishment in, for example, areas of Canada, both Ontario and British Columbia, and so it's established there, and it's causing crop damage, particularly, I believe, to apples in Ontario. So also in Switzerland, for example, it was present for a number of years as more of an urban nuisance pest, but more recently they've started to see crop damage. The pest can have either 1 or 2 generations per year, and it would likely have just 1 generation per year in the UK. But that's not to say that it wouldn't cause crop damage, because in other countries where there's just 1 generation, they are seeing some damage.
Going back to the damage, the level of damage in other countries, do you have the losses in percentage yield at all, rather than in dollars and euros? For example, 30% yield loss.
I don't. I can happily look into that, but I don't have those figures to hand.
Okay, well, something we can back.
Yeah, or my email contact was on the first slide if anyone wants to follow that up with me.
Okay.
Or any other points.
Yes, and if no one's caught that, I can feed back to you as well through my email address that's on the slide.
Thank you.
Is there an EU coordinated response for this pest, and if so, how are we in the UK engaged with it?
There's not sort of one large coordinated EU response at the moment. There's various researchers active in Italy, Switzerland, in Belgium. They're very concerned and are very active in this area also. So there's a lot of research going on. I'm not aware of one EU coordinated response currently. We're certainly obviously keen to learn from the experience of other crop protection specialists and entomologists in other countries, who have obviously got a lead on us in terms of already having experience with the pest established in their countries. So, for example, I've visited colleagues in Switzerland and Italy and Georgia to learn from their experience. So there's a lot of networking that we're involved with in making sure that we're sort of prepared and keeping up on the latest developments.
Great, thank you, Glenn. I think that's probably we've got time for questions at the moment, but we can maybe come back to you at the end if more crop up.
Thank you.
So thank you very much, Glenn. We'll move on to Michelle Fountain and her work on Nesidiocoris.
Okay, so just checking that you can hear me and see the presentation okay, Natalie?
Yeah, looks good, thank you.
Okay, great, thank you. Okay, so I'm going to talk about the project that we had in collaboration with the University of Greenwich Natural Resources Institute, and also Thanet Earth was our industrial partner in this project. And what we aimed to do was identify the sex pheromone of Nesidiocoris tenuis, which is becoming a damaging pest in tomato crops. And in this case, it's the sex pheromone of the female mirid bug. So first, just want to sort of explain why we put the team together that we did. So NIAB EMR and NRI have worked for many years on different insect pest pheromones, and more recently were successful identifying mirid pheromones, and particularly Lygus rubula pennis, which is a major pest of strawberry crops. So from that insect, we identified this sex pheromone, which was produced by the female, but we also managed to find another semiochemical component which acted as a repellent. And we combined these two semiochemicals in trials where we were able to push the insect away from the crop and also pull the insect around the perimeter of the crop into trapping devices. And the good thing about these pheromones is there can be some crossover between species, as has already been mentioned, but quite often they're quite specific to a particular species in a particular environment. And the other good thing, of course, with using pheromones to be able to control insects is that they're compatible with integrated pest management programs. And it's important here to mention that we collaborated also with Thanet Earth on this project, and we were guided by them in terms of the problems that the UK tomato industry would be facing with incursions of this what is normally a biological control, but also we received help in them in terms of field testing the products that we produced. So just as an introduction and a little bit of background to the bug itself, so Nesidiocoris tenuis is actually a tropical mirid bug. It has these probing mouth parts which are normally used for a biological control agent against whitefly, but what tends to happen is when the pest populations decline, the Nesidiocoris switches to feeding on tomato plants. So as I mentioned, it's used as a biological control agent in Mediterranean crops, but in the UK, it's considered as an invasive species. So the damage that you see in tomato crops is you get these necrotic rings around the stems and the leaf petioles, and this can lead to flower abortion, which will obviously result in reduced yields. And there are also reports of punctures on the fruits themselves, which affects fruit quality and marketability. In terms of economic losses, obviously we've got the reduction in yield from the feeding on trusses, but also there's a requirement for pest protection products to be applied, and then you can see a resurgence of the pest insects that were originally being controlled. There's the additional need for monitoring both of Nesidiocoris but also of other pests, and there is the potential for the need to clean the glass houses between cropping cycles, and it's estimated that this can amount to 9.7 million per cropping cycle if we're considering the 180 hectares of tomato that are grown in the UK. So the other really important thing with this pest is that it disrupts integrated pest management. So it's not just the direct damage which is caused by Nesidiocoris. What tends to happen is it's normally a broad spectrum insecticide which is used to knock the populations back in the greenhouses, and we get this reoccurrence of whitefly populations and associated viruses, but also disruption of other biological control systems that might be working perfectly well for controlling spider mites, for example. So in this project, in the 4 months between November and February, we had an ambitious 7 objectives, and I'll just talk through these now. But what I've done in the presentation is there's lots of work that I'm not going to present due to time, so there's lots of work that we did in the background. But initially we obtained and reared unmated Nesidiocoris for the bioassays and for the pheromone identification. We collected headspace volatiles, so this basically means the smell of the insects, and we also did whole body extract collections. The teams at NRI did most of the chemistry, so they're looking at the gas chromatography linked to antennography. And I've got a slide to show you how this works later on. And then later on, there was gas chromatography linked to mass spectrometry, and that was to identify what the actual compounds were in the female sex pheromone. Then once you've achieved all this, then we have to synthesize these compounds, which was also done at NRI, and indeed how to formulate them so that they can be used practically in the glasshouse crops. And then following this, we need to test the efficacy of the attractants. And so we ran a series of laboratory and field bioassays. And I can sum up the laboratory bioassays now in that we actually tried lots of different techniques to get to observe males being attracted to females and also being attracted to potential pheromones, but none of them worked. And the insects wouldn't behave naturally in a laboratory setting. So this is what the insect cultures kind of looks like. So we set these up at NIAB EMR in our insect quarantine facility, and they were given a varied diet, including host plants, and grown up in these larger bug dorm cages as a mass culture. But then as the insects reached their final larval in stage, they were separated into individual boxes so that when they molted through to the final adult stage, that they would be unmated. And what this means normally is that the females are more likely to produce the sex pheromone. So we had at that point individual males and females reared in these individual boxes. So the females can be identified with the ovipositor, which actually they insert into the plant to lay their eggs, and the males obviously do not have an egg-laying organ at the posterior of their abdomen. So this is what the entrainment setup looks like. So when we began to work on the insect, we started with mated males and females because they were immediately available to us, and we entrained these both in groups of males or females and also singly. And then as the unmated males and females started to be produced, we then had individuals and groups of males and females that were unmated, which we entrained. And so what happens here is clean air through a carbon filter passes over the insects and then is captured on a cellulose filter at the other end of this chamber. And we also include a food plant because this helps firstly to keep the insects alive, but also might stimulate the pheromone release from the female insects. So the collections were made at NIAB EMR, and then the antennography was done at the Natural Resources Institute. And this picture on the left-hand side shows the smells that were collected from the female being passed over the antenna of the male. So here we've just got the antenna of the male insect and the head. We don't need the rest of the body for the antenna to be able to detect the female smell. And on the right-hand side is the graphical representation of what is happening in the picture. So along the bottom, this is the smell of the female with all the different compounds that are in there. On the top, there are 2 replicates of the male antenna, so this part here. And the first thing to notice is that males do differ in the sort of magnitude of their response to the smell of the female. So this is why it's important to do replicate runs. But the most important thing to note here is that there's an antennal response of the males to compound A, and there's also an antennal response from the males to compound B, both produced by the females. So at this point, we had 2 peaks from the females that had the potential to be part of the female sex pheromone. So these were then identified on the mass spectra and GC retention times, and NRI then synthesized these 2 compounds, and the synthesized products were again passed over the male antennae, and again the male antennae responded. So that just confirmed that there was definitely those 2 compounds were eliciting a response from the males. So at this point, we thought we had the candidate components of the female sex pheromone. And what was really good was that the structures that of these 2 compounds were different from the pheromones of other species in closely related mirids, including the cocoa mirid, which NRI have also worked on, and also Macrolophus. And as I'm sure you'll understand, it's important that this pheromone is different from Macrolophus because this is used as a biological control agent in UK tomato crops. So once the synthesized compounds have been made, we needed to find a way to release them in a consistent way which was long lasting. So the first thing that was tried were these pipette tips, because this is what we used for the Lygus pheromone, which I talked about earlier, and these worked really well for that species, but they didn't release the compounds consistently for Nesidiocoris. So the team at NRI then switched to these polyethylene vials, and this is what we used in all the future studies for release of the pheromone. And you can see here that NRI changed the release rate by either using pure compound or the compounds in a sunflower oil. But you can see here that these dispensers lasted for over 2 months, and we're releasing the pheromone for at least 2 months. So then finally, we took the dispensers, the pheromone dispensers, and we attached them to these dry glue yellow sticky traps in the greenhouses where we knew there was Nesidiocoris, and we tested either the traps without any pheromone, the traps with a single compound, or traps with both compounds combined. And what you can see here is that essentially the compounds, the 2 compounds combined, captured far more males than either the single compound or the untreated controls. And here you can see the male Nesidiocoris on the trap, and they've actually been ringed so that you can count the numbers that were on the traps. So the project in the short time span was really successful, and these attractants are now available to use in combination with sticky traps to monitor numbers in glass houses and potentially pack houses for incursions. But we still need to do a bit more work to optimize the blend and the release rate of the dispensers, and also how best to combine that with a more optimized trap. And we need to look at what the best trap positioning is in the crop at different times of the crop growth stage. And really importantly, we need to be able to know what the damage threshold is. So how many Nesidiocoris males equals damage to the crop, and that could then obviously trigger the need for a control measure at that point. And then further down the line, and a much sort of bigger question to address is whether we might be able to now start using these semiochemicals to be able to control the insect in the crop. So could we, for example, find an inhibitor and a repellent, as we did with Lygus rudolapennis, to be able to do a push-pull system? Or, as is used with Tuta absoluta, could we look at mating disruption in tomato crops? And what we don't have at the moment is a female attractant. So we've found something which attracts the males, but more work needs to be done looking for a semiochemical to attract the female bugs as well. So I'd just like to finish by thanking all the contributors to this project, especially all the technical staff who, as always, did most of the hard work. And thanks again to the AHDB and BBSRC for funding. Thank you.
Thank you very much, Michelle. I don't have any questions for you at the moment. If anyone could submit them via the box, we can pose them to Michelle. I think maybe people are thinking about it.
Maybe it was so clear, or maybe not.
I thought it was very clear. I suppose in the meantime that we can go back to, oh, here we go. There's a question from Phil. How long will it take to fine tune and implement the technique?
So that really depends on whether we're talking about the technique for monitoring or whether we're talking about the technique for control. So obviously to get to the stage of thresholds will be much quicker than actually a control technique, and there's a lot of legal things that we'd need to address in terms of pest control using the pheromones. But essentially it would, for the monitoring and for thresholds, we'd need to potentially look in a range of different varieties as well. So I mean, it could take a year to 2 years. It's always good to get to repeat these experiments so that you know that you're getting realistic thresholds. So it could be 1 to 2 years for accurate threshold measurements, but much further into the future for control measures.
Thank you. Did you try both compounds on their own?
Yes, we did. Yeah, we did that. I didn't have time to present both, but it was a similar result for the second compound as well.
Thank you. You mentioned optimizing trap design. Are you going to test different colour traps?
That would be part of the trap testing. So I need to say at this point that at the moment we don't have funding to follow on from the project that has just finished, but our aim is to obviously look for sources of funding for the future. And yes, colour, there's obviously you can get an interaction between colour and pheromone, and we found this with Nesidiocoris pabulinus, that in actual fact that species isn't attracted to the same trap as Lygus rudolapennis, and that actually if you have a blue colour, that does increase trap catch. So yes, colour is important to test alongside the pheromone.
Will this eventually be made commercially available, and do you think it will be expensive? He also says, I guess that depends on ultimately the cost of the production of the pheromone. If you have
so from my understanding, it isn't expensive. I think the team at NRI are willing to make these lures available now for people to use in greenhouses and to test out themselves. I don't know whether David Hall has more to comment on the price of the potential price of the attractants.
I've just unmuted you, David.
Okay, and everybody can hear me?
I think so, yep.
Yeah, good. Yeah, well, I mean, obviously it's been unfortunate having to not to reveal the actual structures of the compounds, but if we are looking for further funding, then we do need to keep these confidential for the time being. I mean, well, one is commercially available, one is not actually commercially available, but would be very cheap and easy to make available in this case. And as Michelle says, I mean, in principle, we can supply lures, but I think we want to be sure what we're doing in the next stage, next phase of the project.
Thank you, David. Next question. How many other horticultural crops damaging mirrid species are there in the UK that this technique could be applied to, do you think?
Okay, so that's a good question. So we, well, probably about 8 years ago now, we began working on Lygus rudipennis. We also found the pheromone for Nesidiocoris pabulinus, and also the smaller species, which is a nettle capsid, which attacks, also damages greenhouse crops such as I think it's aubergines and peppers. I can't quite remember, sorry, which species it was, but there are various mirrid species in arable crops as well. And I guess we're always open to new species coming in through plant imports. But in terms of glasshouse crops, I'm not 100% sure about other mirrid species that are damaging to glasshouse crops other than the species I've mentioned.
Okay. Do you anticipate any issues with CRD and licensing?
If we claim if we develop something which is going to be used as pest control, it needs to go through the proper approval channels. And at the moment, that is quite costly, and it would need some commercial backing to be able to do that. So if we embark down that route, it's going to be really important that we have good communication channels with CRD. So at the moment, it doesn't seem to be that easy to do, but I'm hoping that as we're losing approval for more traditional pest control insecticides, that CRD and the approval system might be more open and make an easier route for some of these things to be approved.
Going on from that, another question around that. We've seen how specific these pheromones are, and you alluded to earlier how difficult the regulatory process is going to be for these pheromones as pest controls with a map number. Considering the market size, does there need to be a different framework for semiochemical approvals?
That would be really, really nice. So ourselves and NRI have got a host of different pheromones which we've identified that could potentially be used for control. So the moth pheromones are, they tend to be lumped by the approval system as Lepidopteran pheromones, and that has made it much easier to approve things, for example, Tuta absoluta pheromone. But here we're dealing with a different group of animals, essentially. So either it needs to be made, I think, just insect pheromone based rather than just Lepidopteran, or it needs to be easier to be able to approve groups of insects, so for example mirrid bugs or sawflies, so that we can get these through approval systems much more quickly, but also more cost effectively, because yes, that's a good comment, that sometimes the crops that we're dealing with, they're high value crops, but in terms of the hectarage and the amount of money that the chemical companies can make from them, they're not as lucrative. So we really do need help from CRD and associated bodies to be able to make this easier.
Yes, absolutely. Well, thank you very much for all your answers. Phil's asked, this will have international sales, potentially, I suppose?
Yeah, I mean, I guess that's where it's more lucrative, potentially, in some of the larger crop growing regions. Yeah.
If there are any other questions for Michelle, please do submit them, or for the other speakers. We can spend a couple more minutes. Nikki's asked, for all the speakers, how would you have done this work if funding from this pilot initiative was not available?
Would you like me to go first?
By all means.
Yeah, so I guess because it's directly related to horticulture, the traditional avenue that we would have explored for funding this work would have been AHDB. But as we all know, there's not quite so much money in the pot now for this type of research through AHDB. But it's really important that we get these pilot studies done so that we can apply for much larger projects further down the line. So, for example, this would be taken forward either through an Innovate UK to help a company commercialise the pheromone and how to use it, or we might apply to, for example, BBSRC to do some more fundamental work on the actual biology and ecology of the insects and its relatives in order to understand more about the behaviour. But I think we're at risk of losing that initial funding to be able to get the ideas off the ground. So AHDB funding is really important to us, and this scheme has allowed us to obviously generate pilot data which we can now take forward for both commercial and research bodies.
Shall I go next?
Yes,
can you hear me? Yeah, someone's nodding. Okay, so I suppose the short answer is if we hadn't had this funding, it wouldn't have happened. I mean, it needed to employ someone for 4 months and to get the facility available and so on. And it's the sort of thing that for a pilot project, such funds aren't really available. BBSRC funding is normally for 3-year projects. It is much longer. So it's very welcome to see BBSRC and AHDB working together on this and allowing this opportunity to happen. It wouldn't have happened otherwise.
Thank you.
Yeah, and ditto really for the project that I presented. I mean, you know, with a very specific sort of goal of developing and validating the DNA-based test, that's, you know, not something that we could put to other funding bodies easily for a sort of longer-term project. So I think, you know, on the other hand, the output of having the test and having that as a starting point does enable us to use that and use the information we've learned and the sequence databases and so on to use that as a platform for other funding initiatives. For example, to explore the genetic diversity of the invasive species as it spreads and perhaps as it becomes established within the UK in comparison with other parts of the world. So it's just one angle that we might pursue in the future.
Thank you. Yeah, I mean, I think that's quite a good point to close. Thank you all so much for your contribution. So to James, William, Glen and Michelle, thank you so much. I think it's been a really interesting and useful session. Also, thank you for everyone's questions. If there are further questions, please do submit them via email to me. So that's on natalie.key@ahdb.org.uk, and I'll make sure they get to the right people. Or you can also provide feedback if you have any feedback to give us on how this was presented That's always taken into account for future events. And also to remind you that the webinar recording will be available on the AHDB Horticulture YouTube channel, hopefully by the end of next week. So I think that's everything from us. So thank you very much for listening.